Pain. I'm ready to start. I'm recording the lecture. Let's hope this doesn't get interrupted. I learned one very important thing a few minutes ago. I learned how to turn off my Discord notifications, and I'm very proud of myself. Yay. Yeah, you can laugh, but I didn't know. So I didn't know. That's why you hear all that beep, beep, beep. And then the student who actually used to be a student in this class and then took my other class came and said, Dr. Ladik, if you need help with this, I would be. I was like, yes, please help yourself. So so if you want to help me with technology, please do. I have Brittany who is helping me most of the time and my other two TAs, but I'm taking volunteers all the time. Anyway, are we getting ready? So now we are starting with something way more fun. In my opinion, more fun than evolution. You might find that harder. But from last year's feedback, I improved these lectures. They are way easier now because if I gave you lectures that I planned to give you, it would be crying. Brittany would be happy, but neither of you would. Anyway, Brittany has some great ideas. I can see how you will have fantastic time with a term project. Oh my God, I'm so happy that she's my TA now. This will be fantastic. You will love it. Anyway, we're starting with the unit 2. So we talked about history. We talked about different forms of microbial life were established on Earth and when that happened. And now we're diving a little more into the diversity. So next 4 lectures, like 3, 4, 5, and 6, which means next 2 weeks, will be all about metabolic diversity, which I must be honest is my favorite part of this course, and I find it the easiest one. The harder one is when we come to talk about functional diversity, or when we talk about bacterial diversity, and then you have like 5 different genera that you need to know. This is fun. So might be hard to understand, but for me, I find it fun. And again, I'm, as usual, happy to take any questions. In case you don't know, I'm recording the lecture. Sometimes, if this thing gets interrupted, I will post my lecture from last year because nothing has really changed too much. And when you have some questions, please put it on question forum, written informed units, like unit 1 to whatever forum. So you can post question over there and then I will respond, or I will, if you email me or Brittany, we will respond and then we will post answer on the question forum so everyone can see. And please ask questions. That's great and it's fantastic. Whatever I put in question forum units, it won't go on your tests. So it's just FYI. The only that goes on test is what is in these slides. Only. And yeah, one thing I forgot to announce. You might saw in e-class, there is a review for unit 1 where I had combined slides for lecture 1 and 2. So I did that last year because students asked me main concepts from units, and I do that at the end of every week. So that will help you to study. It's like a study guide. And most of these things I already said, but some people just like to have it. So I will have the same unit same review when we finish metabolic diversity, and then we have our first quiz. I forgot which day it is exactly, but I know it's coming. October 1st. Yeah. Okay. I thought it's September, end of September, something like that. So that will be on your those reviews will be very helpful. And now let's talk about some interesting features of microbial life, which is first phototrophy. And all these chemolithotrophy, fermentation, hydrocarbon metabolism, and anaerobic respiration, we will cover in lectures 4, 5, and 6. But today we are in the area of what means phototrophy. So the word tells you itself, photo light. So that means when organism uses a light energy, so there's the light energy to create some other sources of energy. So it's very common in the world. So it uses a light as a source of energy to start some metabolic process. And Most of you is already familiar with photosynthesis, and many times people associate photosynthesis with only plants. However, microbes were first organisms that did photosynthesis in this world. And we claim that photosynthesis is the most important process in Earth. You might remember why. Who formed oxygen? Photosynthetic organisms. That's how we get the chance to develop in the form we are when the enough of oxygen was accumulated on Earth. So photosynthesis was the most and still is the most important biological process in this planet. So in this process, the energy of light is converted to chemical energy. And simply as it is, organisms that are doing this conversion, that are doing photosynthesis, are called phototrophs. The way how you can memorize that is photo means, of course, light, and troph is a Greek or Latin. I forgot which language is it from. Tropho is like a food, like tropho, like eating. So some organism that is using a light to create its own food. So phototrophs, organisms that are performing photosynthesis, microbial organisms in our case, because we will stay in the area of microbes, are also known as autotrophic organisms. What means autotrophy? So autotrophy means the autotroph is the organism that is able to use carbon dioxide to fix carbon dioxide and to assimilate into the biomass. So basically, organisms that use light and fix carbon dioxide, like plants and some microbes, which we will learn later, are called so carbon dioxide is inorganic carbon, and these organisms are converting it to glucose, other sugars like organic carbon that are later on used for energy. If carbon source is organic, like glucose, for example, and the organism is using a light to start like a metabolic process, is like excitation by light, that organism is called heterotroph. So we would be only heterotroph because we don't do photosynthesis. So that when you hear about photoautotroph, that means organism that use light and fix carbon dioxide. When you hear photoheterotroph, that means organism that uses light as energy, but it is using organic carbon, not carbon dioxide. So back to the photosynthesis, which again, the most important process on the Earth. There are certain requirements that organism has to complete in order to perform photosynthesis, and one of them is the availability of photosynthetic pigments, such as chlorophylls, which are present in plant and algae, and for us, the most important, cyanobacteria. Another type of pigments that are required for photosynthesis that are not part of cyanobacteria are called bacteriochlorophyll. And as the name tells you, bacterial chlorophyll is present in bacteria, so don't get confused. Cyanobacteria are bacteria, but they have chlorophylls. So bacterial chlorophylls you will see in purple and green. And it's actually there is also another word that needs to be used here, and purposefully I didn't put it because we'll talk about that later. It's sulfur. So we'll have green sulfur and purple sulfur bacteria. So those chlorophylls and bacterial chlorophylls, like known as photosynthetic pigment, have very interesting physiology, and they have ability to absorb the photon of light, or simply light, and then they use that energy, like light-derived energy, and convert it to chemical energy, which is ATP, or adenosine triphosphate, and that's where your energy for metabolic processes comes in. But you need to have chlorophyll or bacterial chlorophyll for that. So this most important physiological process on Earth, photosynthesis, is divided in two stages. Stage one, which will be ATP production, where light energy is converted to chemical, and CO2 reduction. And literally the energy that you make, in the form of ATP from light in the phase one, you use to reduce inorganic carbon CO2 and to make sugars that are used to run metabolic process. The energy, as I said, comes in the form of ATP. This is the one that you maybe didn't hear about, or classes, you maybe heard of NADH. NADH, we will hear this a lot of times. It's a molecule that drives energy production in cells. It's like it's energy. The easiest way to memorize is that it is an energy. And the energy from these two molecules is extracted and used for CO2 reduction. And again, that all happens in the process of photosynthesis. So who donates electrons? So what are electrons? So electrons are required to run energy production. So electrons are extracted from high energy compounds like NADH, like glucose, like hydrogen sulfide, like hydrogen. So those molecules are, we say they're highly reduced. It means that they're full of electrons. And some molecule has to have enough electrons that will be extracted. And once when electron is extracted, that electron literally jumps from one to another molecule, and that jumping of electron creates an energy. And we will learn later which molecules can be used. And that's where metabolic diversity comes into the game. Depending on which molecule is available to extract electrons from or to get energy from, the donor, you have different metabolism evolved. So in case of oxygenic photosynthesis, or let's put it on the side, in case of plants and cyanobacteria, and we will stay in the focus of cyanobacteria, the molecule that gives that energy, the donor, so memorize the donor of electrons, is going to be water. And water is the beginning for these molecules, for these organisms. In case of green and purple sulfur bacteria, or let's say green and purple bacteria, the reason why we say it's a sulfur bacteria is because the donor of electrons is hydrogen sulfide. And I purposefully emphasize water and hydrogen sulfide just so I can show you the chemical formula of both molecules. If you can see, they have two hydrogens, they have oxygen, they have sulfur. Oxygen and sulfur are in the same, if you look periodic system, they're in the same column. So hydrogen sulfide can be an energy donor, electron donor. And if you think from a perspective of what was available on early Earth, hydrogen sulfide, what was else available? Hydrogen. So that's why we say that the early metabolisms were organisms that were able to take electrons either from hydrogen sulfide or from hydrogen. That's why they were first. There was no water at the time. Water was present as a vapor. So, but hydrogen sulfide can be used as electron donor. And green and purple sulfur bacteria use it. Because they evolved differently, different branches, depending on the, again, available resources in the earth. And there are three photosynthetic microbes that we will cover here briefly. And again, we'll go in details as we are moving forward through course. Cyanobacteria, purple bacteria, and green bacteria, and I will add sulfur to purple and green bacteria because they are using hydrogen sulfide, and they have very interesting features. So cyanobacteria, as we know, were those guys that created oxygen. As we know, they were pioneers. They created big oxygenation events. So before them, like if we think about 3.5 billion years ago or 4 billion years ago when Luca appeared, Earth was an oxy. But as cyanobacteria start producing oxygen, as we talked, once when oxygen exhausted, oxygen interacted with iron that was in those rocks, so iron 2 plus became was oxidized and became iron 3 plus, and then that was created as a banded iron formation. Once when so cyanobacteria were making oxygen all the time, but the moment when all that iron was exhausted, big oxygenation event occurred. So cyanobacteria set up the basis for our life that we know today. That's why they're so great and so important, and they can be also important from a biotechnological perspective, and we will talk about that as we are moving forward through class. Again, to run this photosynthesis, those organisms have to have pigments. Cyanobacteria have chlorophylls. Green and purple bacteria have bacterial chlorophylls. And these pigments absorb light at different wavelengths. Please don't memorize what's the wavelength for bacterial chlorophyll. Chlorophyll, that really doesn't matter. What it matters is that each pigment has unique absorption spectrum. And chlorophyll is in red and blue light, which means it absorbs red and blue and emits green. That's why you see the green color. Then this is just absorption maximum on these for these spectrums. Again, please don't memorize numbers. There are different chlorophylls, and we will see which. So this is just a picture how the chlorophyll look. So chlorophyll A and bacterial chlorophyll A, as you can see, there is magnesium in the center of it, and the picture here shows the spectrum. Again, really, it doesn't matter at which wavelength. It matters. I would really like if you can remember that they emit green light, which I don't think will be too hard. So, who has this? So, remember, they have different pigments. So, this does matter. Cyanobacteria have chlorophyll. So, not bacterial chlorophyll, they have chlorophyll, and it's chlorophyll A. Some organisms have chlorophyll D, but mainly it's chlorophyll A. That's what it matters. So, some other organisms called prochlorophyll have chlorophyll A and B. But what is more important is what cyanobacteria is. The purple bacteria and green sulfur bacteria have different types of chlorophyll than stone bacteria. Green sulfur bacteria have B, C, D, have Q, D, and E. Purple sulfur bacteria have A and B. And these are important. And this is the detail that I really like to ask, because I do want you to know that there is a difference between chlorophyll and bacterial chlorophyll, and that the first organisms, which are green and purple sulfur bacteria, had bacterial chlorophyll, which was named A and B, or CDE, purple and green bacteria respectively, and chlorophyll A, which is inside the bacteria. So again, by evolving in terms of evolution, by evolving different pigments with different properties, different phototrophs can coexist in the same habitat. So literally, they absorb light at different wavelength because of the pigments that they have, and then they live happily ever after, which is whole point of this beautiful metabolic diversity that we are covering today. And now a little bit more in physiology. So what are reaction centers and antenna pigments? So nothing else but helper pigments that help to collect and harvest all available light so they can get more energy. So, and those chlorophylls and bacterial chlorophyll, think about them as something that is not really floating freely around. So they are usually attached to certain proteins, and then they're stuck into the membrane. And usually they form something called photocomplex. So those chlorophylls and proteins that are forming complex together, they get stuck in the membrane and form photocomplex. So if I ask you what is photocomplex, you will say pigments plus certain accessory proteins. So how many molecules doesn't really matter. Again, I really don't care about numbers, but what it matters is that that photocomplex does have multiple molecules. So it's not only one, it's multiple molecules of chlorophyll and bacterial chlorophyll, and multiple accessory pigment molecules like proteins that help attaching. So they're not just floating around. So that photocomplex, proteins plus chlorophyll or bacterial chlorophyll, they're creating together reaction centers. And those reaction centers are now surrounded by light harvesting pigment. So these are called antenna pigments. And if you think about what antenna is, like, I don't know if you ever had to try to catch a signal with antenna, I did. So they're literally screening for the light. So you have main photocomplex pigments like multiple molecules, accessory pigments like proteins that are helping embedding. And then multiple of these are forming reaction center. And then you have antenna pigments that are surrounding it, and they're screening for the signal. And this is the simplified version. Again, there are variations on the topic, but this is the most simple observation of all photosynthetic complex. So how the photosynthetic membrane actually looks inside. So if you look eukaryotes, for example, you heard about plastids, chloroplasts. So what is chloroplast? Cyanobacteria, right? Chloroplast and mitochondria are actually bacteria. Mitochondria is proteobacteria, chloroplast is cyanobacteria. So they are eaten by endosymbiosis by some host, which we claim it was archaea. And again, that's the last of the theory. So, those chloroplasts or plastids, I will say chloroplasts because that's where main photosynthesis is happening, so there is a photosynthetic membrane that surrounds that cellular material, and it's a part of cellular organs. So this is the picture of chloroplast. And again, the reason why I'm showing it is only because I wanted to go back to the endosymbiotic theory, what actually chloroplast is. So in chloroplasts, chlorophylls, like pigments, are attached to the membranes, and those membranes look like a sheet, like they are stuck on each other, or they can be vesicle. Those are called thylakoid membranes. So these stacks of membranes are called thylakoids, and they're nothing but they're built like cell membrane, lipid bilayer, and multiple thylakoids are called grana or granum in Latin. And the inside of the chloroplast, like it would be cytosol bacteria, is called stroma. The area in between thylakoids is called thylakoid area. And this morphology, so again, the only reason why I'm pointing out this morphology, and I do want you to have that in your minds, is because this is actually bacteria. This is what started. This is what started endosymbiotic theory, which goes back to our first unit. And diving into the prokaryotes, so they do not have chloroplasts. So again, in plants, you have chloroplasts. But if you think about microbes like cyanobacteria, purple and green microbes, they do not have chloroplasts because they do not have organelles. But they still have something very similar, like you can make analogy, because internal cell membrane of prokaryotes is making like cellular invaginations, and that's where chlorophylls are integrated. Again, just because they do not have organelles. So the same principle, chlorophylls are embedded there with proteins. And now, analog to the thylakoids, those membrane arrangements are called chromatophores. And chromatophores are like vesicles. And the lamellas are also membrane invaginations, but they have a different morphology. So lamellas are like a stack. So in my opinion, lamellas more remind on thylakoids, where chromatophores are like membrane vesicles, which are a little like a circle, like just a different morphology. But you need to know the difference between chromatophore and lamella. That is important. Of course, as usual, there is exception, and those are cyanobacteria. They have thylakoids again, just because they are very similar to chloroplasts in plants, and I would say they are chloroplasts in plants. So cyanobacteria actually have thylakoids. They're same called thylakoids, but purple and green bacteria have lamellae and chromatophores. And in addition to chlorophylls and bacteriochlorophylls, there are other pigments like carotenoids, phycobilins. Again, just there to get different wavelength of light depending on what is available. And carotenoids are very famous nowadays in biotechnology because they have antioxidative properties. So whenever you buy that sunscreen, it will say, oh, it has carotenoids because it protects from sunlight. So think about that. Why were they important in ancient Earth when there was no light protection? And again, talking about carotenoids, later in course, we'll talk about their application in biotechnology. So we got this from microbes. Carotenoids actually are photoprotective agents. That's why they are in sunscreen. So they bring light harmful to cells, and they can catalyze because they can catalyze photooxidation. And so when you have that a lot of light, what can happen during that photooxidation, you can get production of that singlet oxygen. I just need to put this as a superscript 1, and oxygen 2 goes in the sunscreen. because this singlet oxygen, like that's produced from a lot of light, it can oxidize photocomplexes and make them non-functional. And carotenoids are protected. They literally cleanse that toxic oxygen species. You probably heard about that reactive oxygen species, or ROS, and they prevent harmful photooxidation. Again, this is really exploited to the sunscreens. But they have to, so photosynthetic organisms must live on light. They need light for their energy processes. Whenever cell evolves these carotenoids, that means it has extra light protection, which means that when there is too much light, it can develop this protection, and it can still harvest light for energy without damaging its oxidation, without damaging its structure. Phycobilly proteins, cell proteins, and they're organized in phycobilisms. So phycobilly proteins are another pigments in addition to carotenoids. So they're not main photosynthetic pigments, and they're aggregated together in the structure called phycobilisms. And again, this might come to the exam where I ask you to fill up the sentence. Again, spelling doesn't matter, but you need to know what it is. Those arrangements of phycobilly proteins, called phycobilisms, are attached to the membrane, and they can facilitate energy transfer to reaction centers. So they're there to literally transfer energy, because there are some cyanobacteria that grow at lower light intensities. So they need extra molecule that will pick up all available light and bring it to the reaction center where it can be converted. Now, back to the first organisms that actually started everything, which would be purple and green phototrophic bacteria. And I will start with the purple bacteria. And these two groups of organisms actually have very specific reaction centers or photosystems. And if you combine two of them together, you get cyanobacteria and plants. But these guys were first. That's why that is the reason why I'm teaching them in this way. So purple bacteria are anoxygenic. What means anoxygenic? That means they do not produce oxygen. What that means is that the donor of electrons, the energy source, is hydrogen sulfide. So not water, it's H2S. And then when they oxidize it, they produce hydrogen sulfide, they make sulfur dioxide. Unlike cyanobacteria, this makes oxygen. Now, the reaction center in these bacteria, it's called P870. Again, number really doesn't matter so much, but what it matters is how this center looks. So it is called quinone based, unlike the other one, which has another molecule associated. So what you have here, and you will see again, please don't memorize all these molecules. What matters are these quinone A's and B's, but most important quinone, because this center is called quinone base. And the quinone is the most important molecule in this reaction center. And we will go in details when we talk more about photosynthesis. In green bacteria, reaction center, again, number doesn't matter. So it's lower number, it's 840, but what matters is that it has pyridoxine. So it's pyridoxine based. So if you compare with this one, where quinone is the place where electrons get accumulated, this one has iron-based center and pyridoxine, which is another electron carrier. So again, we will talk in details when we come to the diversity of bacteria. For now, only know that purple bacteria have quinone-based reaction center, while green bacteria have pyridoxine-based reaction center, and electron donor is the same for both, is hydrogen sulfide, and they are anoxygenic. And this is what matters. The source of reducing power is hydrogen sulfide, and this very interesting feature that these organisms have, because when hydrogen sulfide gets oxidized, like when it releases electrons, very often it can create sulfur globules. what that means, it creates like an elemental sulfur. And if it's a green bacteria, if it's a green sulfur bacteria, those elemental sulfurs are created outside of the cell. In case of purple, they are inside the cell. So why that matters? Well, because that elemental sulfur is now a storage. In case they run out of hydrogen sulfide, which can happen, and you can think about in ancient Earth, that probably happened multiple times, they can still take that sulfur because it's still full of electrons, it's not still exhausted, and they can take energy from that. So they can take that energy and still reduce CO2 and make their organic compound. So it's the way of, it's like a saving for a rainy days. And they do that quite commonly. Oxygenic photosynthesis, which is cyanobacteria, that's what we care about, or plants, which is later on. Those two quinone-based and ferredoxin-based reaction centers are combined together, and we have them as photosystems 1 and 2. Again, numbers don't matter. What matters is which one is which. So this is called, if you ever saw this, called Z scheme of photosynthesis, and photosystem 2, which surprisingly comes first. In my head, everything functions from left to right. So this is where everything starts. That's why I think like kids. So this one is quinine based. So I do want you to point out that this photosystem 2 is quinine based, which is again coming from purple bacteria. And then there is all that electron transfer that's happening. So light is here, electron gets excited. And then there is electron transfer from different quinones, because quinone can accept them and then release them back. And then it comes to the cytochrome. Again, don't memorize that. What is important to memorize that electron that now got transferred from quinone, this is quinone pool in photosystem 2, is going to be transferred to another photosystem, which is, guess what? Ferredoxin base, which comes from green sulfur bacteria. So this is where reduction power is going to form in photosystem 1. So again, photosystem 2 comes before photosystem 1. And as you also know, there is an exception, there are exceptions. So in oxygenic so sometimes anoxygenic photosynthesis, which we know is happening in green and purple sulfur bacteria, can happen in cyanobacteria, which are known as oxygenic photosynthesis. Why and how? So if you have oxygenic photosynthesis, where water is electron donor, where oxygen is produced, the one that made oxygen on Earth, those two photosystems that are combination between purple and green sulfur bacteria function as a tandem, like they operate together. But what can happen often in nature, that the first in line, which is surprisingly photosystem 2, can be blocked. Something can happen, mutation or something blocks the initial photosystem. In that case, only photosystem 1 is available. So if only photosystem 1 is available, the only way how cyanobacteria can survive is to take something else other than water and extract electrons from it. And of course, it will use either hydrogen sulfide or hydrogen as energy donor, electron donor. So this is adaptation that is very often in this organism, which I love to ask, not to write, but to recognize. It's called oscillatoria linetica, and it's this is the organism, and it is cyanobacteria, but it actually does anoxygenic photosynthesis and deposits those as elemental sulfur granules, and it's really common in salt ponds, so you can always enrich for that organism. So it's considered to be cyanobacteria, but it's actually depositing sulfur. It's using sulfur and deposit granules more like a sulfur, green sulfur bacteria. This is common example that I really like, because again, not to write, but to recognize. I really don't care about spell, so don't bother about that. Bother about understanding. So again, from evolution standpoint, this process of, we call this cyclic photophosphorylation. So why I'm saying cyclic? Because it goes in cycle. What means photophosphorylation? So to explain it briefly, to get energy, you need to phosphorylate ADP to get ATP. So that's why we called it phosphorylation. Usually in organisms that are not light consumers, you will get that phosphorylation in something called the electron transport chain in mitochondria. And again, the point is to phosphorylate ADP to get ATP, because then you use ATP to run your processes. In this case, this will be photo, like light-based phosphorylation. So this process of photophosphorylation in both anoxygenic and oxygenic phototroph is just one of the many indicators of their evolutionary connection. So connection between anoxygenic green and purple sulfur bacteria and cyanobacteria does exist, and this photophosphorylation and photosystems, which I mentioned, are great example of it. So what is the first evidence? So the first evidence is, of course, cyclic photophosphorylation, like use of light to make ATP and later on NADH. Another evidence is that similarity between purple sulfur bacteria and green sulfur bacteria reaction centers and photosystems in cyanobacteria and later on in plants. And you can see that, so you will have purple sulfur bacteria that have quinone-based system. So the same quinone-based system you will have in cyanobacteria, but that will be photosystem 2, P680. Then you will have at the same time with purple bacteria, you will have green sulfur bacteria, which again now have only photosystem 1, which is pyridoxine based. But guess what? In cyanobacteria, you will have that other photosystem as a part of integrated Z scheme. And then all of that now exists in cyanobacteria. And you see all of that later on in plants, which are again result of endosymbiosis when host ate like cyanobacteria, which became chloroplast. And that's that, like you see why we in cyanobacteria, we call that stack of membrane silico, it's same like in plants. So that's that evolutionary connection that we really, really want to understand. And it's similar, in my opinion, very easy. So this is what we now know, that purple and green sulfur bacteria lived on Earth much before cyanobacteria. So they were they were early metabolism organisms. And later on in this class, actually today, we will start with some organisms that were even before phototroph organisms, but they had to live without oxygen. That's why they evolved to utilize hydrogen sulfide or hydrogen and to make even additional electron depositories just so they can survive unwelcoming conditions on Earth. So this anoxygenic photosynthesis was actually the first and key photosynthesis on Earth that helped in development of oxygen. Cyanobacteria's key invent was connecting those two photosystems and evolving the ability to use water as electricity. So again, talking about conditions on early Earth, there was no organic carbon. Only available form of carbon was this inorganic carbon dioxide, which is the reason why fixation of inorganic carbon, which we claim to be autotrophy, is one of the most important early events on Earth. So you probably heard about carbon dioxide fixation when we talk about plants, and we also talk about cyanobacteria as organisms that can fix carbon dioxide. But there are other ways to fix carbon dioxide other than the one that is very famous in plant that we call Calvin cycle, and we will go in details now. So this carbon dioxide is highly oxidized, like form of carbon that must be reduced, must be converted to organic form in order to form it, put it in the cell material, into the biomass. So it's not like carbon dioxide is not really nice in terms of energy. Like in order to fix that carbon dioxide, to make it available for cells, you have to put a lot of energy. So it's energetically unfavorable. So all autotrophs, which are organisms that can fix carbon dioxide. So those are phototrophs and chemolithotrophs. What are chemolithotrophs? Again, will be mentioned during the course. So phototrophs use energy of light to reduce this carbon dioxide. So they do all of that during photosynthesis. Chemolithotrophs are not using light. They're using inorganic carbon. So not glucose, something other than glucose. And later on, we will learn about hydrocarbons or even something that is not really usual for energy source, which you will probably figure it out. So very often organisms use molecules as energy source, but as a carbon source, they use carbon dioxide, and they need a lot of energy. And there are three modes, actually four modes that are used to fix carbon dioxide. Three of them I really like to ask. One of them is very old, and I don't really put a lot of effort to emphasize it. But the first one is Calvin cycle. And again, please don't memorize all of these processes. Like in my other class, I ask all of this. For you, it's important to understand that the Calvin cycle is the most distributed process in nature, and it's present. You need to know in which organisms it's present. Of course, in cyanobacteria, it's most efficient. Well, actually, it's not most efficient. It's very efficient, but it's not most efficient. Sorry. It's present in purple bacteria, and it's present in some chemolithotropic bacteria, but most important that I want you to know is that it's present in cyano and purple bacteria. I do want you to know two key enzymes of Calvin cycle. One of them is Rubisco. You heard about Rubisco? Yep, everyone does. Riboso bisphosphate carboxylase, and the other one is phosphoribulose. It's enough to know Rubisco and dual function of Rubisco, which we will cover later on in class. So Rubisco and phosphoribulose are operating in these structures that are called carboxysms, and they're not organelles. They're structures inside of cyanobacterial cell, because bacteria, microbes, do not have organelles. So they do have like a polyhedral structure, so they look like organelle, but they are not. They're not same organelles like we have in cell. And the reason why cyanobacteria evolved carboxysms is simply so they can concentrate carbon dioxide, and just to facilitate that assimilation. And then in those structures called carboxysms, they accumulate this Rubisco, because Rubisco does carboxylation and oxygenation and is actually really required for carbon dioxide fixation. In carboxysm, this carbon dioxide is trapped and it's ready to be run through the Calvin cycle. And the other reason why you need to have, why organisms had to evolve carboxysms, is because Rubisco is, so carbon dioxide is one of the substances for Rubisco. The other substance is oxygen. But that alternative substance is not really doing any good to the cells. So this is the way to restrict the access to the second substrate. And that's, so basically that's the evolutionary reason why carboxysms have to do that. So green sulfur and non-sulfur bacteria, which again we will cover later, but for now know only that there are sulfur bacteria. They do fix carbon dioxide, but they don't run Calvin cycle. What do they run is something called reverse. So they have different ways to fix carbon dioxide. And there are three other ways. That hydroxypropionate pathway under number 2, which I don't really ask, but I do clearly ask reverse TCA cycle and this acetyl coenzyme A or Wood-Langdale pathway, which is probably the third one is the most cool way to fix carbon dioxide, and it's very ancient. Reverse TCA cycle, as the name says, is just, if you heard about TCA cycle, cycle. This one just operates in the back. So the same how rubisco and phosphoribulose are key enzymes for Calvin cycle, which are those that I want you to know. For reverse DCA cycle, again, you could know that it's in chlorobium, but more important for me is to know that there are two key enzymes that are two peroxidase enzymes. Again, do not memorize all these steps. This is not for this purpose of this class. It's just for people who want to learn more. And in other class, I ask all these questions in detail. But the most important, those peroxin linked to 1 and 2 enzymes, and they operate in some hyperthermophilic archaea, which are thermoproteus sulfolobus or Aquifex. Aquifex is something that I usually like to add, but know that it operates in hyperthermics. Again, hydroxypropionate, just as a set of side information, it's found in oral plastics. It's one of the earliest, and again functions in hyperthermophilic archaea. What does that tell you? If something functions, what means hyperthermophilic? That means something that loves high temperature. So that means it had to evolve in early Earth when temperature was very high. And last but not least, acetylcoenzyme A or wood-Langdale, which is present in archaea that are called methanogenic or acetogenic archaea. This one is fantastic because it's very old and it's the most efficient. So Calvin cycle is the most abundant, but this one is the most efficient. And it's not even a cycle, it's a more linear, but we say it's cycle, that I just make up like a lapsus. So methanogenic archaea and acetogenic archaea. So what means methanogen? Methanogen is something that produces methane, hydrocarbon. At the end of this unit, we will learn about these organisms. But what is important for this pathway to know is that it is most efficient, and the C enzyme is carbon monoxide dehydrogenase, CO dehydrogenase. And as you can see, electrons are used from hydrogen. This is a good indicator that tells you that this was the first pathway that formed on Earth. Why? Because hydrogen was overly abundant. And these organisms that produces methane actually use hydrogen to reduce carbon dioxide, to fix carbon dioxide via this pathway. And this would be this. So we, as I promised, we finished earlier. If you want, we can go over unit 1, or you can go home and we can talk about this later on. I would suggest go home